Hello, good morning everyone. My name is Anjali Jaiswal. I am pursuing MSc Food Technology from Institute of Agriculture Science, Banaras Hindu University. Today's my presentation is on the topic clinical testing of nutraceuticals and health foods. Before I proceeding to my slides, first of all I would like to aware you about what is nutraceuticals. So the term nutraceutical was coined by Doctor Stephen D. Phillips in 1989, who was MD founder and chairman of the Foundation for Innovative and Medicine (FIA). According to Dr. Phillips, nutraceuticals is any substance that is a food or a part of food that provides medicinal or health benefits, including the prevention and treatment of disease. Means that at our daily basis we used to take some nutrients for the development and the metabolism of the body, but nutraceutical is that thing, is a that substance who's providing us nutrients along with medicinal or health benefits. So it is a very you can say that it is a very good thing that we are getting two things from a one or from any one substance. So let's proceed. Health Ministry of Canada defined it as a product isolated or purified from the food generally sold in medicinal form, not assisted with food, and demonstrated to have a physiological benefit and provide protection against chronic disease. As we know that tomato. So tomato is a food in which we have lycopene content. So after the extraction of lycopene content from tomato, it is now called as a nutraceutical. And lycopene has very much its protection against, like it is a very much anti-carcinogenic, and it is very much immune modulatory and rapid junction communication. It helps in many mechanisms. So reduces the, it also reduces the cardiovascular disease. So it helps in many forms. Another definition of nutraceutical is, a nutraceutical is any substance considered as a food or a part which, in addition to its normal nutrition value, provides health benefits including the prevention of disease or promotion of health. As I said earlier, it is any non-toxic food component that has scientifically proven health benefits including disease treatment or prevention. It must be non-toxic, because if we consumed it and if there will be a toxicity, so definitely it will get harmful to the to every consumers. So it must be non-toxic. The functional component of the food must be standardized in the nutraceutical product and produced under good manufacturing practices. Whosoever the nutraceutical is, wherever nutraceutical is produced or manufactured, it should be in a way like good manufacturing practices, because at last we have to consume that. So it must attain all the good manufacturing practices throughout the process. So it is a picture that I have inserted that claims that what is nutraceutical and what is food supplements. So clinical evidence is much important in every new product development, because at last it will be consumed by us. It will be consumed by humans. So it must be fully treated with every single test, everything, with a proof, with a written data, with a written information, everything, so that in future if something will be happen against this product, so we can recheck or we can modify it by the previous updating information. So it is very necessary to have a clinical evidence of everything. So what clinical evidences is required of nutraceuticals? What evidence exists? What conclusions can be made? And what are some needs and opportunities for evidence generation in nutraceuticals? Means why we need clinical evidences? Why does all are important? So in the US, evidence requirements for nutraceuticals include evaluation of safety for ingredients which are required to be listed on the label. All the things which are in the product should be listed on the label, that how much carbohydrate, how much minerals, how much vitamins, everything should be labeled in a proper way and in proper with proper accuracy. It is optional to make claims of clinical benefit to bring a nutraceutical to a market. So how a nutraceutical product is developed? So it is a I have a flow chart in the right side. So there is a new nutraceutical development, how we develop a new nutraceutical product. So here is a health condition identification. Okay, food matrix selection. It is very much necessary that what the base we are going to use. What is the base of that product for which we are going to use? So after that, food matrix and relevant compounds identification in the phytocomplex. What are the compounds in that base, in that food matrix which we had selected, and what is their functional activity, biological activity. After that, there will be a vitro safety assessment test. It is done to make sure about all the above things. Then metabolic profile study, relevant compounds characterization, then assessment of mechanism of action of the phytocomplex and relevant compounds involved. After that, many clinical trials and a proper dose assessment, everything is done, and then market availability, that now my nutraceutical product is properly gone through all the tests and now it is prepared to launch in the market, and after that only the nutraceutical product can be launched. So what are the significance or what are the aspects to develop a successful nutraceutical product? So first is the purity and sustainability of components. Second, scientific evidence of effectiveness. Third, guarantee of product quality. Fourth, the comfort and convenience of the formulation. Means everything should be there. It is a mandatory for any nutraceutical development that it must have pure form, and scientific evidences should be there, because we after means nutraceutical is something, or any food is something which cannot be fit for every consumer. So and scientific evidence helps that in future if anyone claims that your product is not good, yeah, uh, your products harms me or anything, so we have scientific evidence that we have tested. We have gone through many tested before launching it. So that's why we have proof of everything, because we have scientifically experimented and everything proved, tested. So every record has to be maintained. Guarantee of product quality. Quality must be good, because quality is the main aspect for the food for every human consumption, because quality is the main key for any food to be consumed. The comfort and convenience of the formulation should be very in the comfort way and the convenience. So first of all, when we thought of developing a new nutraceutical product, first of all we go through the microbial testing, that what kind of microbes are present there, or what like bacteria, viruses, fungi, anything can be there in the food product. So we have to go for testing, microbial testing. The contaminated food can lead to food poisoning, and to ensure the food safety, the analysis of food is extremely important. So food poisoning, if a food is contaminated, it definitely goes to be to lead food poisoning. And food safety is the first and major aspect of any food before consumption. So it must be ensured that the food is safe for the human consumption. So food contamination, how food contamination occurs? It mainly occurs due to viruses, bacteria, metals or chemicals. Food poisoning is generally caused due to the bacterial contamination. And the major classes of bacteria that cause food poisoning are E. coli, Staphylococci, Salmonella, Clostridium, Campylobacter, and Shigella. They can cause symptoms like they can cause nausea, vomiting, diarrhea, dehydration, distress, and may in severe case may also death. Hence, microbial testing is very much important. So some tests are there. First, we have a bioburden test. So bioburden test is defined as the total number of viable microorganisms on a medical device, and then use the number to determine the appropriate parameters for its final sterilization. Means in this test, we generally measure the total number of bacteria living on a surface, and that has not been sterilized. We have to sterilize them in this process, because in any product, if means if bacteria growth in any product, if there is bacteria, they growth very rapidly, they grow at a very high rate, and they can cause many severe problems in after consuming by humans. So it must be sterilized before. After that, total aerobic microbial count. This test determines the total number of aerobic bacteria per ml or gram of the personal care, cosmetic, or pharmaceutical products. So, this test determines the total number of aerobic bacteria per ml or gram. For this test, the incubation periods is generally for from 48 to 72 hours, and the total aerobic microbial count is a mandatory test for every lot manufacturer of health, beauty, and personal care products. So the next is total combined yeast and molds count. This In this test we use Aspergillus brasiliensis and Candida albicans to determine the total number of yeast and fungi per ml or gram of the personal care, cosmetics, or pharmaceutical products. In this method, we have to give 5 days for incubation, and this method, total combined yeast and mold count test, it is a mandatory test for every lot manufacturer of health, beauty, and personal care products. Next is testing of nutritional value. So, nutritional value is very much important as we take any food just because of nutrients available in that food. or particular product. Nutrients are the basic things of our life by which we can survive. So nutritional value of any product is very important. So the nutritional value of food refers to the quantity and quality of nutrients found in the food item. Yes, yes, it refers to both quantity and quality, because certain limits or certain intake daily intake is there in the standards. We have to take according to that. So everything has a limit. That's similar in the case of nutritional value. Yeah, nutrients. We have some limits of taking of intaking. So, information about the energy measured in calories. The macronutrients like carbohydrates, proteins, fats, and micronutrients, vitamins, and minerals. And beside this, phytochemicals of the foods are required to understand this all procedure. Next is the laboratory-based nutritional analysis. So how we are going to analysis the nutrients available in the product. So laboratory-based nutritional analysis can be used for virtually any food type. This method can be used when general nutritional labeling needs, validating manufacturer supplier nutritional information, validating data generated from software-based database analysis, product contains unique or exotic ingredients or processing techniques, analyzing, analyzing food for animals, pet foods and agricultural foods. Accurate nutrient per 100 gram values are needed for database inclusion. So in this point, it is so it is very much that things that if we want to measure any nutrient, so we have to take at least 100 gram of that sample or that any product, in which we are going to test that how much just suppose for an example I want to know the content of carbohydrates. So in that case I will be reporting that this much of carbohydrate is found in, yeah, is analysis or is this amount of carbohydrate is present in 100 gram of sample. So this type of data should be prepared. Reports will feature values per 100 gram and per serving size of the product. So after this, where applicable, percentage of RDI, that is recommended daily dietary intake, and second DV percent, that is daily values percent, will also be provided. मतलब this RDI and DV values both are important. Additionally, voluntary nutrients, vitamins and minerals are available. So what we are going to analyze in the laboratory based on the nutritional aspect. So there is protein, moisture, ash, fat profile, in which fat profile contains a lot of things like total fat, saturated fat, monosaturated fat, polyunsaturated fat, or trans fat, cis fat. Many types of fat are there. So every analysis should be done. Sugar profile, total dietary fibers, sodium, calcium, iron, cholesterol, vitamin A, vitamin C, carbohydrates, calories, and calories also from fat. Next is heavy metals. So it is very important to test heavy metals because heavy metals have some, not some, they have potentially toxicity. If we consume in much higher amount, it may it may cause severe problems to our body. So heavy metals should not contain much limit in any product. Uh, so here we go. A heavy metals panel is a group of tests that measures the quantity of specific potentially toxic metals in the blood, urine, or more rarely in the hair or other body tissues or fluid. Heavy metals, metalloids, any metal species may be considered a contaminant if it occurs where it is unwanted or in a form of concentration that causes a detrimental human or environmental effect. Means heavy metals uh, cause contamination in both cases, in human and in environmental case also. Metalloids include lead, cadmium, mercury, arsenic, chromium, copper, selenium, nickel, silver, and zinc. So, heavy metals contamination from vegetables. So, as we know that vegetables are the life of our surviving. Means it is a very important for our life because it is our first diet that we have. Vegetables are the major source of our every nutrients. So if we have uh, heavy metals, having heavy metals on the surface of vegetables, they are going to, and we consume that vegetables, definitely they are going to uh, cause a health risk. So, vegetables are rich sources of vitamins, minerals, and fibers, and also have beneficial antioxidative effects. However, intake of heavy metals contaminated vegetables may pose a risk to the human health. Heavy metal contamination of food is one of the most important aspects of food quality assurance. As we know that heavy metals are non-biodegradable and it also contaminates the environment. So, in case of vegetables, they are deposited on the surface, and the tissues of the vegetable absorb that heavy metals, and then if we consume, it get harm to us. Calorific value. So, calorific value of the sample should be analyzed. Calorific value can be performed on a variety of fuels including biomass, oils, coals, and wood. For this, a weighed quantity of the sample is burned in the presence of oxygen in a bomb calorimeter under controlled conditions. And then the heat of combustion is calculated from the weight of the sample and the rise in temperature. Means from both aspects uh, the heat of combustion is calculated. After this, the value which we have obtained is the gross heat of combustion at constant volume, which is expressed as megajoule per kg. Nutraceuticals label claim testing. So it is one of the key ways of ensuring label claims compliance and overall quality of product is rigorous testing. So, with the current GMP standards, quality testing is an indispensable part of the manufacturing process. Testing is required to ensure that nutraceuticals, nutritional supplements, and dietary products meet their label claims. Whatever is mentioned on the label should be met with the products, with the inside the package. Means the product containing the things, the nutrients which are claimed on label. So for this measurement, accurately measure, HPLC is very much effective in measuring vitamins, minerals, and other additives present in raw materials and finished goods. So now the how the nutraceutical products are certified, under what condition, under what organization they are going to be certified, and after that they are going to launched in the market. So AgMark, as we know that AgMark is stands for agricultural marketing. It is a quality certification mark of the Government of India given to products conforming to scientifically laid down quality standards. Products commonly certified under Agmark are like honey, ghee, butter, turmeric, curry powder, cardamom, cumin, coriander, vegetable oil, spices, wheat, pulses, and many more as per the requirement by the customer. All the products as per required by the customer all have agricultural marketing certification. So how the certification of authorization takes place? It is given to the packers, authorizing them to grade their commodity under AGMAR. So the packers are allowed to get the AGMARC uh, logo or certification under their commodity. Period of validity will be 5 years. The validity should be of 5 years. For granting new certificate of authorization, separate application for each commodity should be produced. Means a separate application should be prepared to get all this authorization. Documents which are going to be attached with the application are sketch of the premises, what the premises, under what premises you are manufacturing that product, declaration regarding proprietorship, partnership, etc., ownership of the premises, ownership of trade brand label, use of good grade quality containers for packing commodities, means what packaging we are use for our product. Then a copy of license from municipality, bank reference, letter from the bank regarding the transaction to the packer with the bank, list of machineries, specimen signature, मतलब all the specimens persons should have their signature on the papers, medical fitness certificate of employer. So next one is Bureau of Indian Standards, which we call as BIS. So it was the act which protect the interest of consumers, operates a product certification scheme. Under this scheme, the license are granted to the manufacturers who are capable of producing goods on continuous basis as per relevant Indian standards. Like we have food business operators, they can usually apply for this. Testing being necessary adjunct to product quality evaluation, the need for making available required testing facilities arises. It is this context that BIS has established a network of 8 laboratories of its own in the country to cater to testing of samples generated from its product certification scheme. So there is a laboratory advisory committee. It is the one of the 6 statutory advisory committees of the BIS, and the BIS advises on policy matters relating to establishment and operation of its laboratories related matter. And laboratory advisory committee members are the members of. So the FSSI also have so the after the product development there must be the logo or authorization from the FSSAI also. The FSSAI stands for Food Safety and Standards Authority of India for laying down science-based standards for articles of food and to regulate their manufacture, storage, distribution, sale, and import to ensure availability of safe and wholesome food for human consumption and for matters connected therewith on incidental thereto. So next is packaging and labeling of foods. No person shall manufacture, distribute or sell or expose for sale or dispatch or deliver to any agent or broker for the purpose of sale any packaged food products which are not marked and labeled in the manner as may be specified by regulations. Means that provided that the labels shall not contain any statement, any claim, design, or device which is false or misleading in any particular concerning the food products contained in the package or concerning the quantity or the nutritive value implying medicinal or therapeutic claims or in relation to place of origin of the said food products. Second one is every food business operator shall ensure that the labeling and presentation of food, including their shape, appearance, or packaging materials used, the manner in which they are arranged, and the setting in which they are displayed, and the information which is made available about them through whatever medium does not mislead consumers. Basically, it was saying that. Anything if we have mentioning on the label should meet all the means that means anything that is labeled on the labeling portion must be there in the product. This simple as this line contains. Next is, so now I have attached a research paper on a randomized double-blinded placebo controlled clinical study of the effects of a nutraceutical combination on LDL cholesterol levels and lipid pattern in subjects with suboptimal blood cholesterol levels. So before I go through this research paper, want you to I want you to confirm or aware about you what is the single blinded, double blinded, and placebo control. So a single blinded is that, for example, if I am going to check the effect of a drug on a person, and I know that I am giving this drug for this effect on the person, and the person in front of me is not knowing about what is the product, yeah, what is that which I am going to give him or her. So he or she don't know. So this case is called single blinded. In this case, one person is knowing about the fact that this is a drug and it will going to affect someone. So in double blinded, in this case, both the giver and the consumer both don't know about the drug which we are going to study. So after taking and after that the result came, then after we know that what is that drug and what is now the effect of that particular drug. And in placebo, so placebo is a substance that is not medicine, but that is given to someone who is told that it is a medicine used to test the effect of a drug or to please a patient. So in this research paper, uh, the nutraceutical is combined with the nutraceutical is combined with the uh, red yeast rice standardized to contain 5 mg of monoquilins from Monascus purpureus, also the niacin 27 mg and polycosanols 10 mg. So this treatment is statistically shows the significant difference between the treatments in favor of the combined nutraceuticals, and also it is used to improve the combined nutraceutical group only. No significant changes in other clinical and laboratory parameters were observed. So the trials was well tolerated and significantly shows that the plasma levels get reduced. So here are the references from which I have taken that information, and thank you.